Okay, so good afternoon, everyone. And this class today concludes our discussion about bacterial growth. And I remind you that you have a test on Wednesday, and the test consists of all material in this particular chapter on growth, okay, up till the end of today's class. This means this is a really circumscribed amount of information that you need to comprehend. It's not a great deal of kind of conceptual stuff in this, unlike metabolism and cellular respiration, etc. So this is a good chance for you to do really well on your test. And tomorrow, Tuesday, 12 o'clock, I will be having a live session for you to be able to ask me questions to help you with your revision. Okay, so let's proceed and get this chapter finished. So we're talking about bacterial cell division. Bacteria, with very rare exceptions, divide by binary fission. Let's just remind ourselves about what that entails. Just 1 second, and let me see if I can get this to work. No, can't. There we go. Okay, so that's simple enough. One thing that I will mention, and that is that when the cells divide, they don't necessarily separate completely. They are separate. They're separate because they have entirely separate cell walls and cell membranes, but they may in fact remain loosely attached to one another, often by a capsule or whatever. And that is what gives us our characteristic filaments, chains, or in the case of streptococcus, chains of cocci, staphylococcus, bunches all stuck together. So the cells don't necessarily fly apart from one another. This here is of two cells which have just about completed dividing, but it's a nice picture because it shows you here, you can clearly see the plasma membranes, the cell membrane of each cell is now entire, but the cell walls are not complete yet. So the two cells have not yet separated completely. And the cell wall manufacture will continue all the way down and all the way up until the cells separate completely. Okay, so we now come to some important kind of mathematical considerations. Because cells divide by binary fission, they're doubling their number each time they divide. So the time that is required for a division to take place is called the generation time. And it varies tremendously. It varies with the species of bacterium that you're talking about. It varies with the conditions that the bacteria are kept under. And environmental considerations may be very, very important. For example, temperature has a tremendous effect on the rate at which bacteria will divide. So they have an optimum growth temperature at which they divide with the greatest efficiency. In the case of an ordinary bacterium such as E. coli, for example, if we're growing it in culture, then we can expect at optimum temperature, which is about 37 degrees Celsius, with a rich nutrient medium, we can expect a generation time of about 20 minutes. It's it can be extremely rapid. It depends on the bacterium. Some bacteria have a very long generation time. One which, in particular, which is famous for the length of its generation time, is Mycobacterium tuberculosis, the causative agent of tuberculosis, has a very long generation time. But something like E. coli can be very rapid. So binary fission is doubling the cell number in case of E. coli every 20 minutes or so. We can work that out mathematically by looking at this simple little formula expression here. 2 to the number of generations as a power will give you the number of cells that result from a division. So let's have this has some important implications. When we look at the growth of bacteria, we're looking at very rapid development of very large cell numbers. And as a result, we usually try and express these logarithmically on a log scale. So let's have a look here. We're going to consider a single cell dividing. By the way, this is incorrect. 2 to the 0 is not 1. But in any case, we'll ignore that for the moment. Let's have a look and see. Look, our little formula, 2 to the power, right, is the way that we're going to work out how many cells there are per generation, starting with 1. So we start with 1 cell, and 5 generations time, it is 32, right? Because that's 2 to the 5. 10 generations time, 2 to the 10, 1024. 2 to the 15, etc., etc., etc. If we are trying to represent this on an arithmetic scale, it'll be a curve, but that curve has a very particular characteristic, and that is if we take the log of the numbers, we discover that we can express it as a straight line there. So here is this period of growth. We're watching the number of generations, right? 5 generations, 10 generations, 15, etc., 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 etc. Go back here, and you'll see the numbers. There they are. If we represent it arithmetically, it's a curve like this. But if we represent these numbers as the log of the number, we see that it has this very characteristic straight line. And we call this period of growth the period of log growth. It's the log, also referred to as the log phase of growth. And it is one particularly interesting time in a culture, in the life of a culture, because it's a time when there's very little cell death taking place. And instead, the whole population is reproducing at its optimum generation time. And the generation time stays constant throughout. They are unaffected by environmental influences such as the amount of nutrient or the temperature. The temperature is kept constant. The nutrients are abundant. They're going to reproduce at their optimum rate. And it produces this characteristic log phase growth, which we see as a straight line on a log plot. So what is happening is that here the population is reproducing exponentially. If we look at the whole, if we take a flask of nutrient, let's say, and we put an inoculum into that nutrient, shake it up, and then we monitor the growth of bacteria. We monitor the increase in numbers of bacteria in that culture. We see a very characteristic pattern of growth. There are actually, we recognize 4 particular stages. A lag, they're called lag phase, log phase, which should be familiar to you already, because that's our period of optimum growth. Stationary phase, and then the death phase. So we start off this, think about this for a minute. I take my nutrient flask, I inoculate it, disperse the cells, and just leave it. If I watch that for some time, there's going to be very little change in the number of cells. This is the typical behavior during lag. It doesn't mean to say that nothing is happening. There may be all sorts of things happening. The cells, for example, may be growing in size, but not dividing. They may be replicating their DNA, but not yet dividing. We're not going to see an increase in cell number, but all sorts of metabolic things may be happening. The other really important thing about lag phase is, do you remember what we said about quorum sensing? The bacteria can actually talk to one another. They, by means of releasing substances which are picked up by other bacteria as signals that other members of their species or whatever are present in the environment. And it also cues them to what those other members are doing, whether they are yet dividing. The lag phase is an important period for quorum sensing. And as it gets to its end, often it's because the population of bacteria have signaled to one another to begin dividing. And when they do so, they very rapidly enter the next phase, which is log phase. And that is what we've described here. That is this period here, when the generation time remains optimal and constant. There's no death of cells, or very, very little, so little that you wouldn't notice. And instead, the population number increases logarithmically. That continues for a certain length of time, but then environmental conditions begin to take their toll. First of all, nutrients starts to become limiting. Second of all, we may have all sorts of other things happen. They may be using up all the oxygen in the environment, for example. You may be accumulating waste. You may be accumulating waste like acids and all sorts of things in the environment. All of these things begin to have a detrimental effect on the population. And now the average generation time is no longer optimal. It begins to lengthen, but in addition, we enter a phase now where cells begin to die. There is a lengthy period when as many cells are produced, die. So the population remains constant. And this is called stationary phase. It may be quite lengthy. Stationary phase may last for a long time. And it's a very important phase because a number of other things begin to happen in stationary phase. And for example, if we were growing a gram-positive bacterium, such as Clostridium, or something like that, it is during stationary phase that those cells will begin to make endospores, will begin to produce endospores. That is a specialized form of cell division, which results not in the production of an equal daughter cell, but of a small cell enclosed in a spore coat, which is highly resistant to adverse environmental conditions. So there may be some very important things happening during stationary phase. If these adverse conditions continue, then we reach a point where cells begin to die exponentially. And this is called the death phase. It's where the loss of cells now is happening logarithmically, and the population begins to crash. Various things can happen during death phase. If you've just got a batch culture and you forget about it and it enters death phase and it accumulates masses and masses of waste products and there's no nutrient left, etc., 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 then you may well kill off your entire culture. that is quite possible that the cell number eventually goes to 0. But very, very often we find that there are cells produced which are able to resist these adverse conditions. Very often they become metabolically depressed. Their metabolism is reduced. Very often their size is reduced. But they are able to persist. We call them persister cells. And these persister cells may, in fact, be very important for rejuvenating the population when conditions are optimal again. It's these persister cells which feed the new generation. So this growth curve, the bacterial growth curve, is very, very important because this helps us to understand the behavior of our cultures when in lab, et cetera. But it's also just think about the bacterial growth curve in terms, for example, of infection. When we're talking about a pathogen, for example, which infects a new host, we'll see much the same kind of behavior, a lag phase, a log phase, ultimately probably enter a stationary phase, if not begin to die off. This death phase here can be very strongly influenced. For example, if in an infection, this death phase can be produced or amplified rather by things like the activity of the immune system operating on this population. What is the immune system doing? It's making the environment even more hostile to the population and speeding this death phase. So there are all sorts of implications of this growth curve, of the bacterial growth curve in all sorts of things, in food production, in industrial production. I'll give you another example. I told you that the stationary phase is really important. And for example, in industrial usage of bacteria to produce products, if we're growing something like Clostridium, which produces many useful solvents and things like that, is often here in stationary phase that those are produced in bulk. So in that instance, we may be wanting to create conditions where we maintain our population in stationary phase as long as possible so that we can get the products that we need. Okay, just hold on one sec. Okay, so um, in the laboratory, we very, very often want to know what our bacterial populations are. There's a singular difficulty about this, and that is that in many situations, like in a batch culture or something like that, the numbers are absolutely enormous. Per milliliter, you may have billions of bacterial cells, and this in itself presents something of a problem for counting them. There is an opposite problem as well, and that is in some environmental situations, bacterial numbers may be so low that it is very difficult to count them. And that's very often the case, for example, in fresh waters, in lakes, streams, this sort of thing. Bacterial counts may be extremely low. It actually becomes difficult to find them to actually see what the total population is. We're going to look at 4 different ways in the laboratory that we actually count bacteria. 3 of them I'll do in detail. That's plate count, filtration, and the direct microscopic count. This one here I will just mention, the most probable number count, the most probable number method. But these 3, plate count, filtration, direct count, these you'll need to know something about. Okay, so first of all, one of the commonest standard ways of counting bacteria is to especially prepare an agar plate for counting. And we'll discuss the methods that we use to do that. But I'll just tell you that statistically, what we want to head for is a petri dish, a plate, agar plate, that has between 30 and 300 colonies on it to count. And here's problem number 1. If I have a batch culture, I have E. coli or something like that, I want to know what the total population per milliliter in that is, then if I plated a milliliter onto a plate, the whole plate would be thick with bacteria. I wouldn't even have colonies. I would have the whole thing covered with a sheet of bacteria growing confluently. I wouldn't be able to count between 30 and 300 colonies. Here's the assumption. When we do a plate count, we assume that each colony visible grew from one bacterium. We often refer to that bacteria as a colony-forming unit to reflect the fact that we accept that occasionally we may have 2 bacteria stuck together which grew to form the colony. So, but we assume that our colony-forming units is a reflection of the population of bacteria in our original sample. Well, if we've got billions of bacteria in 1 milliliter, what is our stratagem for plating so that we end up with between 30 and 300 bacteria on the plate forming those colonies? The answer is that we have to take a very, very, very small volume of bacteria. And to take a very small volume, first of all, you can do that physically with a pipette, just taking a smaller and smaller volume. But a much simpler way is to dilute your sample. And what we do is we do a serial dilution of the original sample to the point that when we take a subsample of one of the dilutions, we put it onto a plate, we have sampled between 30 and 300 colony forming units. So there are 2 ways to produce that plate, and we'll discuss both the pour plate and the spread plate. Okay, so for the moment, we're going to go through our serial dilution series. Remember that the aim of the serial dilution is to end up with a very small subsample of your original volume. Okay, here's our original. We've got a batch culture of something, let's say E. coli. What we're going to do is we're going to take a subsample of that. We're only going to take 1 milliliter, but that milliliter may contain billions of cells. we were to just plate that 1 milliliter, we would not form colonies even. They would grow confidently and you wouldn't be able to see anything. So instead, we take 1 milliliter and we put it into 9 milliliters of broth. So there's 9 milliliters of broth there. And 1 milliliter plus 9 is 10 milliliters in total. We have made a 1 milliliter in 10 dilution. A 1 milliliter in 10 dilution. Now, I tell you right now, if we were to plate a 1 milliliter of that, of our 1 in 10 dilution, we would still have far too many colonies to count. Let's get the math straight in our heads. 1 milliliter into 9 gives you a 1 in 10 dilution. That is a 10 to the minus 1 dilution. 10 to the minus 1 is 1 in 10. Okay. So we got far too many colonies there. We've got to do a higher dilution. We're now going to take 1 milliliter of our 1 in 10 dilution. We're going to do the same thing. We're going to put 1 milliliter into 9 milliliters of broth. This is a 1 in 10 dilution of a 1 in 10 dilution. You multiply them. 1 in 10 and 1 in 10 gives you 1 in 100 dilution of our original. So that is 1 in 100. That is 10 to the minus 2. Do it again. We're now going to take 1/10. We go to 1 mil into 9 mils. That is 1 in 10 of a 1 in 100. That is 1 in 1,000. 10 to the minus 3. So on and so on. We usually try and extend our dilution series way beyond where we need it to be. So when we look at our place, never mind for the moment how we do this, we're just looking at the plates. We see, what did we say? We wanted between 30 and 300 colonies on a plate for it to be statistically viable. This one, there's too few. That is a 10 to the minus 5 dilution. Here, this one is probably also just less than 30. So that one also is too dilute. We don't use that. But here, this plate here has between 30 and 300. And we count them. Okay, so this plate here, we'll stick with their count. They say there's 54 colonies here. There's 54 colonies, and it is a 10 to the minus 3 dilution. How do I work out what my original was? Well, I just take that 54 and I multiply it by the reciprocal of the dilution. So it's 50, what I have is 54 colonies times 10 to the 3. 54 times 1,000. 54,000, 5.4 times 10 to the 4. Colony forming units per milliliter. That is the proper designation. Okay, this dilution series here, we have gone through to 1 in 1,000. We plated 1 mil and we ended up with 54 colonies. So we had a 10 to the minus 3 dilution, 1 in 1,000. So we have the reciprocal of 10 to the minus 3 is 10 to the 3. So we've got 54 times 10 to the 3 is equal to 5.4 times 10 to the 4. And we express it as colony forming units per milliliter of the original. It's relatively simple. There is a complication, though, which you must keep your eyes on, especially in lab, okay, when you do these dilution series. 1 milliliter is quite a lot to inoculate onto a plate. For various practical reasons, it is much more useful to have a smaller amount. So let's just take 0.1 mil. Okay, I'll just do it with the first plate and you'll see what the effect is. This, we had 1 mil in 9, that's a 1 in 10. And if I take 0.1 mil of that, I have to take account of that. And it becomes a 10 to the minus 2. So it is 10 to the minus 1. 0.1 mil is 10 to the minus 1 mil. So it's 10 to the minus 1 times 10 to the minus 1, it's 10 to the minus 2. Remember that the size of your sample here can be in itself a dilution, because you're only taking part of the sample. So you take 0.1 times 0.1 ends up 10 to the minus 2. All right. So this is a dilution series. And here are our plates. This ended up being the one that we counted. I should tell you that you normally, if you're doing a dilution series like this, you normally do several duplicates of this. And you average your count. It's a much more statistically viable way of doing it. And this is a standard and very good way of assessing how many viable cells, how many living cells do you have per milliliter in your original sample. Okay, so we just need to spend a minute just thinking about how we actually prepare those agar plates. There are two ways to do it. And the first way is to actually take your, let's go to this one that was our most useful one. The first way is to take that milliliter sample there and actually mix it into the liquid agar, which you're going to pour into this dish. That may sound a bit extraordinary, because agar, after all, melted agar is hot. But don't forget, agar melts at 100 degrees Celsius, but it only sets between 40 and 45 degrees Celsius. And between 40 and 45 degrees Celsius, most bacteria can withstand that for a short period of time. That's not going to hurt them at all. So what you do is you take that 1 milliliter subsample from your dilution series, you put it into an empty Petri dish, and then you pour your melted agar onto it, and you make sure that you mix it really, 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 really well. The agar very soon sets. Don't forget you're pouring your agar cool, so it very soon sets. The bacteria are suspended and in that agar, nutrient agar mix, and they eat. Hopefully, you've diluted sufficiently that you've spread your colony forming units out apart from one another. So most of them will be single cells. Those single cells will each grow and they will form colonies. When you do a pour plate, you will observe that some colonies grow on the surface and others are embedded in the agar. It doesn't make any difference. They're still easy to count. So that is called a pour plate method. The other way, which is probably more common, is that we do a spread plate. And we use an already prepared agar, nutrient agar here. And we put in our subsample, if we were doing here, we took 1 mil, and we put that onto the surface of the agar. And we then spread it out. We use a spreader. If you can see here, looks, there it is there. A glass, it's usually a glass spreader, easily sterilized. And you spread the liquid out. You carry on spreading until all of the liquid is absorbed by the agar. And hopefully all your bacteria are spread out, each colony forming unit separate from another. And then you incubate that. And you come back later, the next day or whenever, and you will observe this sort of situation with between 30 and 300 colonies on the plate. That is ideal. Again, remember, we usually duplicate several times in order to get a really accurate count by spread plating. This is a very quick, easy method to use. Important, though, that you know exactly what your dilution series entailed, so that you have an accurate measure of the dilution, because that is what enables you to produce a workable plate. You work back to your original count. Okay, now here's another situation. This is a situation often in environmental working especially with fresh water. Marine systems are not as bad. Marine systems actually have quite a high bacterial population, but fresh water is often very, very, very low. And it becomes difficult to get an accurate count. So what we often do then is that we will pass water through a filter so that we can put a large volume of liquid through and we can extract the bacteria. We use a filter which is small enough to stop the bacteria, to filter bacteria out of the solution. Once we've passed the water sample, obviously a known volume through the filter, the bacteria will be trapped on the surface like this. And we can then we just take the filter and we plop it down onto a petri dish, onto an agar, nutrient agar in a petri dish like this. And we now allow it to grow. And it's the same idea that what we're counting, we try to get between 30 and 300 colony forming units. We know the volume that we pass through. We can count on the plate like this. And now we can work backwards to our original volume and work at how many per milliliter, how many colony forming units per milliliter. There's just one other thing I should mention with this. This exact method is also used generally for doing things like counting how many fecal bacteria are in water sample, that sort of thing. And what in those cases, very often what you'll do a filtration, but you might have many, many, many different kinds of bacteria that you're not interested in. Okay. And instead, what you do is you take your filter and you put it down onto a highly selective medium, like McConkey's or something like that, so that only the bacteria that you are interested in grow. So you can take a filter like this and strongly select for particular bacteria of interest that might be fairly rare in the environmental sample, even if the total numbers of bacteria are very high. The bacteria you are interested in may be fairly low in number. You take, again, you do the filtration, take the filter and put it onto a highly selective medium, which will give you a count for the bacteria that you are particularly interested in. Okay, I'm just going to mention this method, the most probable number method. I don't think that in the course of your microbiology career you'll ever use it, but it is used especially in environmental microbiology, and it's worth just knowing about it. It's in a way a sort of dilution method. What we're going to do is we're going to inoculate multiple test tubes with decreasing volumes of the environmental sample, and we're going to decrease that volume until we are not actually transferring any bacteria at all. So in other words, you can do it through a dilution series or whatever, but ultimately you will dilute to the extent that you have no bacteria present. So I'll show. It doesn't make much sense without me showing you exactly what it's done. You have to set up multiple tube sets. So normally you do 5 per dilution. So all that you need to know is here, what we're doing is we're effectively doing a dilution series. Here they're doing it simply by adding decreasing amounts of inoculum. And what they would do, they'd set up tubes 5, 5, 5. And below here, there would probably be another set of 5. So this would be 0.0. The next one down would be 0.01 milliliters. And when you then you incubate all these test tubes, the whole dilution series, and you look to see Where is the dilution series where I have no growth at all? In this, it would be like, this is 0.1, 0.01, all the tubes would remain sterile because you haven't picked up any bacteria at that high a dilution, at that low an inoculant size. In other words, the same thing. You haven't picked up any bacteria in this series here. You go to the first series where you do see any growth at all, and that is 0.1 milliliters. There is one tube that grew. Only one of your tubes was inoculated with a bacterium at that volume. This one here, you're inoculating with 1 mil. 3 tubes had bacteria in them. 3 of the 1 mil inoculations. 1, 2, 3. 10 mils, at a 10 mil sample, all of them did. 1, 2, 3, 4, 5. And so you go, nothing, take the next one up. 0.1, then the next one up, 1 and 10. That is a standard dilution series for most probable number. And now there's a lengthy mathematical theory behind all of this, statistics and et cetera, we're not going to talk about, because all you do is you use a most probable number table. Have a look here, this standard dilution series, we got 5, 3, 1. 5 grew, 3 grew, 1 grew. That gives us a number, a code number in a way. You can think of it, 5, 3, 1. You look it up on the most probable number index. Here it is here. How many positives did you have? 5, 3, 1. And this now gives this mathematically works out for you a most probable number per 100 milliliters in your original sample. And this tells us 110 cells per 100 milliliters. In other words, this is a method which you can use for very dilute samples, for very rare samples. It is extremely useful then. And it will tell you 110 per 100 milliliter. And it also gives you a statistical confidence. It says there's 95% confidence that your population is between 34 and 250, with the most probable number, 110. It's a tedious method, but very, very useful, especially in situations where there is a small population of bacteria that need to be counted. Okay, so our next method is to actually look at the bacteria themselves, count them under a microscope. This is not easily done. For a start, many bacteria are motile, so they swim vigorously. It's really difficult just looking through an ordinary microscope and trying to work out how many bacteria are you seeing in a field. It's very, very difficult. The second thing is, under normal circumstances, making a microscope slide, the volume you are looking at in the microscope field is difficult to determine, because just picture a microscope slide, a sample, aqueous sample, and then a cover slip on top. The cover slip is, in fact, kind of floating on the liquid underneath. It is difficult to tell what is the volume that you're looking at. So instead, we use a specialized microscope slide system. It is called a Petroff-Hauser cell counter. And basically, it consists of a slide which is scored with lines and which has raised edges that lift a rigid cover slip a set distance away from the slide, so that when you look through, you are looking at a constant volume of liquid underneath that microscope slide. The slide is scored with squares that you can observe, and you can count how many bacteria are present in each of the squares. and that you have an area that you're looking at, a known volume, because there is a set distance between the slide and the cover slip. So you know the volume, and you can directly observe the number of bacteria in that volume. It's a very quick, very easy way to determine the cell population in a set volume. It does not require incubation. You can just sample directly. It means you get your results immediately. You don't have to incubate a plate in an incubator for days in order to get the colony count, etc. So it's a very, very simple method. There are some problems with it, as I said. The first is bacteria are motile. This is what you observe when you look through this system. Here it is here. These are actually raised. I know it's an optical illusion. This here is actually raised, and it describes a set volume between a set distance between the side surface here and the cover slip surface. So there's a constant distance, and you're looking at a set area which is described by these squares. So what you're observing is a set volume, and when you here, you can observe here. Have a look here, and you'll see the slide. The slide is here, and there are slightly raised edges which lift the cover slip, which is rigid. By the way, it's not thin like normal cover slips. It lifts it a set distance away from the slide, and underneath here you have set areas. So when you're looking at this, what you're looking at is a set volume. You can work out exactly what the volume is, for example, for that square that you're looking at there, and you can count the bacteria 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's fine as long as they're not motile. If the bacteria are swimming, it becomes very, very difficult. And very often, if you're using a motile bacteria, you'll actually kill the bacteria first before you do this. This has another advantage. You can move. You can count a large number of squares. For instance, you would count that probably there. For this population of bacteria, you would count that there. Then move to another one, count again. Then move to another one, count again. Then move to another one, count again. And the set volume is here. It's standard for all of these manufactured Petrov-Hauser counting chambers. And the volume in that large square is over 1,250,000 of a milliliter. So you know that it is 1.25 times 10 to the minus 1, 2, 3, 4, 5, 6. 1.25 times 10 to the minus 6 milliliters are contained in that there. So you've got the number of bacteria, you've got your volume, and you can work out then the number of bacteria per milliliter. Okay, so here's the last method that we use for assessing bacterial populations. This is very often done if we are tracking a batch culture and we want to be able to somehow measure bacterial size, bacterial population size over time. We'll very often use a spectrophotometer, a spectrophotometric method in order to do so. The principle is really simple. If I take a tube of nutrient broth with no bacteria in it, I can shine a light through it, and that it will absorb a certain amount of light and it will transmit a certain amount of light. If I then inoculate it with bacteria, the presence of bacteria in that nutrient broth is going to reduce the amount of light that is transmitted by the broth. Some of it's going to be scattered. Some of the light is going to be absorbed by the bacterial cells. But the net effect is the more bacteria I have in tube, the less light is going to be transmitted. So I used a machine called a spectrophotometer, and that's exactly what it does. It has a light source, and it has a light sensor. And we put a tube in between it, and we measure how much light is actually transmitted through that tube. What we'll do in performing this kind of experiment, we will use our nutrient broth with no bacteria as a blank. We use that to set 0, because we have to accept that the nutrient broth on its own is going to absorb some light, but that absorption will stay the same. No matter what bacteria are doing, it's going to stay the same. So we want to use that to set a 0. We then inoculate. We spread the bacteria, and we can follow periodically. We can measure how much light is passed through this tube. As the numbers increase, the amount of light is going to drop. The amount of light transmitted is going to drop. And we're going to be able to plot that absorption on a curve. And if we plotted the absorption on a curve, we would observe much the same as we saw originally in our growth curve. We would see that there's a period of lag, a period of log, a period of stationary, and then a decrease afterwards as entered death phase. So that's fine. We can do that. We can just plot it out. We refer to the absorption that the solution does as being the turbidity. So we are measuring the amount of cloudiness, the turbidity, and we get this kind of growth. Okay, on those curves. There's a problem, and that is how do we relate that statistic, the turbidity, how do we relate that to an actual cell number? That's a problem, right? Because it's all very well to follow the turbidity and the changes in turbidity. It reflects what's happening in the population, certainly, but it doesn't tell you what the population is unless you have prepared what is called a standard curve before you even begin to use the spectrophotometry methods routinely in your lab to follow particular culture behavior. You perform a standard curve experiment. And so what we do is, first of all, we take a method where we know how to count the number of cells. Let's just say we do a standard plate count. Okay, so here we're going to follow a culture. We're going to grow a culture as normal. We're going to inoculate. We'll see a lag phase. Then we'll see a log phase. And then we'll see a stationary phase. So as you follow this here, you're plating as you did before. You're getting your population counts there. At the same time, you are doing it with your spectrophotometer, and you're getting a turbidity of the solution at each of these population sizes. And what you end up with is a curve, a standard curve, where you have how much light is being absorbed by the turbidity, and you have a measure of the number of bacteria per milliliter. The number of bacteria per milliliter determined by some population method. You could also do direct. You could be counting these in a Petrov-Hauser chamber, for example. You have some measure of the actual population size, and then you have a measure of the turbidity at that particular population size. And it gives you how much light is being absorbed by how many bacteria, and you have a standard curve. Now, you don't have to repeat the spread plating, whatever. Now, as long as you observe the same conditions of growth and everything else, you can just measure the turbidity, and you can measure turbidity, you can measure the absorbance of light, and will give you a particular population size. So in actual fact, you can look up how to grow certain bacteria. It will tell you exactly, grow them in this medium at this temperature, and you can use this particular standard curve. You don't have to do the standard curve. Things for well-known bacteria, E. coli, these sorts of things, these standard curves are well established as long as you observe the same conditions in the experiment. And I think this is actually wrong, sorry. I have sent an email to you giving you for tomorrow, Tuesday, giving you the time and the meeting ID for a question time. Your test on Wednesday is going to be on the growth chapters only up until the end of today. Okay. And with that, we'll say goodbye for the moment, and I'll see you hopefully tomorrow.